First of all, thanks to the organizers for inviting us to conference on gold nano in Lyon. We belong to the University of Bourgogne-Franche-Comté at Dijon. Today, we talk about the role played by the gold nanoparticles. How can we use the gold nanoparticles to determine the structures of protein using an optical spectroscopic approach? SERS, the Surface Enhanced Raman Spectroscopy. Understanding the interaction between protein and gold is of fundamental interest. Indeed, when nanoparticles are immersed in a biological medium, a layer of protein called corona is formed on its surface. But what are the conformations of these proteins? What are their structures? Let's take the example of the BSA protein, the bovine serum albumin. It becomes a standard to investigate the protein at the nanoscale. This protein, in its native form, can be spherical, globular, or more precisely as a heart shape. Protein conformation is very sensitive on its physiological environment, such as the temperature or the pH. The protein can elongate also in the shape of a cigar in its F form, and under even more harsh conditions, the protein can also be denatured. Our goal today is to investigate how does the surface act on the protein structure. Conformational changes in protein extend over a large time window from femtoseconds to several seconds. Amino acids constitute the primary structure of the protein. They are the fastest mobile elements with vibration at the femtosecond, rotation at the picosecond, and torsion of the side chain at the nanosecond. This motion has been studied extensively by dynamic molecular simulation. The secondary structure is constituted by the association or the arrangement of the amino acid into the alpha helix and beta sheet. Fluctuation of this domain appear in the time window between the micro and millisecond. The tertiary structure is responsible for the globular shape of the protein. Folding and unfolding of protein must be monitored in the time range of the second. Scanning probe microscopy is particularly well suited to monitor this conformation at the single protein level. Denature, cigar, and globular shape are here directly imaged. Studying the secondary structure, several methods have been developed. First, nuclear magnetic resonance spectroscopy is the most used method. It's a powerful tool for investigating the structure and dynamics, but it requires a high protein concentration, a large infrastructure, and long measurement times, which makes studies difficult and expensive. X-ray crystallography allows for atomic level structural information, but crystal structure of protein can differ significantly from its native structure in aqueous solution. Optical spectroscopy, such as circular dichroism in UV range, has been developed to require relatively smaller amounts of sample under physiological conditions. This has resulted in its widespread use by the communities in biochemistry, structural biology, and pharmacy. However, the UV spectrum provides a limited resolution on structural information. A spectrum needs fitting procedure and deconvolutions. In infrared region, due to the strong optical absorption of water, analyses are usually performed in dry medium. Spectra can overlap, which makes the signature difficult to recognize. By a monochromatic excitation in the visible range, Raman spectroscopy seems to provide more detailed information on the vibrational modes of the secondary structure. Nevertheless, the inelastic light scattered by molecules still requires a large amount of protein mass. The signal must be enhanced using plasmonic substrates. We will focus then on this method called Surface Enhanced Raman Spectroscopy, the SERS. The electromagnetic enhancement necessary to Raman scattering can reach 10 to the power 8 when the molecules are in close vicinity of gold nanoparticles. This opens up to detect very small amount of molecules, including single protein. Let's compare the composition of the secondary structure from the results given by the different techniques. We observe some discrepancy between the content of alpha helix in the structure. The circular dichroism underestimates the percentage, whereas classical Raman overestimates the presence of alpha helix. Concerning the beta structure, Raman and infrared spectroscopies deliver a high degree of information about the different levels. The beta turn random coil can be discerned from beta sheet. One can then ask the question if one can determine by an ultra-sensitive spectroscopy like the SERS the structural composition of single protein. This was our challenge. Our SERS setup is based on an homemade confocal microscope. The excitation line is a 785 nanometer laser source, and we use a water immersion objective of high numerical aperture 1.2. Two optical arms enabled to detect simultaneously the Raman spectra every 100 milliseconds and the Raman scattering using an avalanche photodiode every few microseconds. The plasmonic substrate is made, as you can see, of an assembly of close packed gold nanoparticles of 100 nanometer diameter. A microfluidic PDMS cell is capping the plasmonic substrate. We use it to control the volume, the flow of biological reagent. The temperature of the cell is regulated from 20 to 16 degrees Celsius within 0.1 degrees Celsius using 4 Peltier elements. The BSA protein was diluted in the PBS buffer at pH varying between 2 and 7. The single molecule regime was reached when the BSA concentration is less than 1 micromolar. Indeed, at this concentration, the distance between proteins is 3 times longer than the size of gold nanoparticles. Here is a successive series of single protein spectra during about 15 seconds. You can observe very thin Raman lines. At first sight, the spectrum does not appear globally to be very fluctuating. The most interesting Raman band to examine the structure is called the amide one, lying between 1600 and 1700 cm1. It arises in large part 80% from the backbone CO stretching vibration and CN group 10 to 20%. The exact band position is determined by the backbone conformation and the hydrogen bonding pattern. The zoom on the amide one clearly shows that the conformation fluctuates. Fluctuation is more visible as a Raman shift than variation in the Raman intensity. These frequency shifts are responsible for the broadening of the Raman band. See here the superposition of 90 spectra. The advantage of having spectra of single molecules is to be able to analyze the statistical distribution of the position of the maximum peak. The position of the top of each peak is detected by a red dot using a MATLAB software such as FinePeaks. Statistically, red dots are mainly centered in few bands. The most populated band, here shown in blue, is around 1620 cm1. It corresponds to the presence of alpha helix. The other band in red matches the beta sheet. Two other bands stand out and reveals the random coil and the beta turn structure. The accuracy of the distribution will depend on the number of spectra. Here, 20,000 spectra are processed. This is the way, by counting, we measure the percentage in helix and sheet in a structure. You do realize the advantage of counting. This avoids the curve fitting. Not only the detection of single protein provides results similar to other approach, but also we have access to information on conformations that are very difficult to get using other techniques. This allows us to complete the picture on the composition of the secondary structure. Typically, beta turn or random coil type conformation have only been determined before by Fourier transform infrared spectroscopy. This method could be applied to two other spectral windows, named amide 2 and then 3. Amide 2 derives mainly from in-plane NH bending, around half of its potential energy. The rest of the potential energy arises from the CN, about 30%, and the CC, about 10%, stretching vibrations. Amide 3 are more complex bands as it depends on the details of side chain and hydrogen bonding. Therefore, these bands are usually less used for the extraction of structural information. Nevertheless, the fluctuations are considerable and symptomatic of the conformation changes. We observe identical Raman shift here. It confirms the composition of the alpha helix percentages. Nevertheless, the amide 1 remains the most interesting spectral window because it provides more information. We were interested in varying the environment of the protein, first by lowering the pH. No conformational change is observed down to a pH of 4.5, but below, the profile of the spectrum density changed. From this, we deduce that the alpha percentage in the structure decreased by half. Indeed, above the isoelectric point of the protein is in its native globular form. The structure is getting the shape of a cigar down to a pH of 3.5. Finally, at low pH, the spectrum density appears more flat, and this is due to the composition in helix, which gets below 45%. The protein seems then in its denatured form. The protein is then unfolded in the goal surface. To understand these conformational changes, it is necessary to look at the type of bond during the absorption of the protein onto the goal. The thiol goal bond can be monitored by the vibration band center at 385 cm1. This binding, now classified as physisorption, appears much more stable than the secondary structure of the protein. At a physiological pH 7.6, the thiol binding is monitored for approximately half of the spectra when the protein is detected. In addition, it turns out that the interaction by thiols is quite stable above the pH 4. The interaction seems reduced slightly when the protein was found to be denatured. Protein can also interact with gold via its amino acid in its surface. Amino acids are mostly detected using the amide 3 and also the window between 4000 and 1500 cm1. At pH 7, the phenylalanine, the tyrosine, and the lysine were the most detected amino acid. At pH 4, tryptophan is even more marked than lysine. At pH 2, the spectrum changes totally. Only the amino acids which are hydrophobic are detected, such as leucine, cysteine. These hydrophobic amino acids are normally located in the core of the native protein, which is a sign that the protein is denatured. These interactions can be explained from an electrostatic point of view by considering the isoelectric point of both the protein and the gold surface. The isoelectric point of BSA is 4.7. This means that under this value, the electric charge of this protein is positive. Our gold nanoparticles were stabilized using citrate capping, colored here in blue. Citrate is negatively charged when the pH exceeds 4. Therefore, the native protein in heart shape will not be attracted by the citrate surfactant. Indeed, a substitution of the citrate by the BSA is assumed. The BSA at physiological pH is attached to gold using the thiol group and its most negatively charged amino acid. The situation is similar at the lowest pH. The protein is electrostatically repelled by citrate. we saw that the thiol bond was less active. The protein being denatured, it becomes hydrophobic and it prefers the gold surface rather than remaining in solution. The intermediate situation of the cigar shape remains the most complex. The protein structure is the most fluctuating, and from an absorption point of view, this is the less favorable situation. In fact, the citrate agent can act as an electrostatic bridge between the protein and the gold. The orientation of the protein is no longer fixed here. This supports the detection of rare amino acids in the protein such as tryptophan, but whose Raman cross-section is the strongest. Now that we have understood the effect of pH and surface, it is interesting to follow the evolution of protein conformation with temperature. The protein remains in globular shape at room temperature. We identify this conformation with a composition of the helix well above 50%. This is what we saw in the pH study. The spectrum counting statistics show that the Raman scattering is then predominant at 1625 cm1. As the temperature increases, the percentage of helix tends to decrease. We observe a slight plateau at 50%, where we find the characteristics of the cigar shape already observed in pH. In this shape, the Raman scattering of the alpha helix is shifted here to the high energy. Beyond 40 degrees Celsius, the helix composition decreases linearly to reach rate below 40% we set when the protein was denatured. From a spectroscopic point of view, the helix again have a less energetic Raman scattering. We can explain a loss of energy in two ways. First, from a chemical point of view, by a charge transfer between the molecule and the plasmonic surface, either because the optical index around the particle due to absorption increases locally from 1.33 to 1.4, and this has the effect of shifting the resonance toward lower energies. In the case of a temperature change, the electrical charge are not modified. We observe that the proportion of thiol bond remain unchanged. The observed shift of 8 cm1 are therefore due to an index variation. We find the result with the temperature seen with the pH. That is to say, the protein in cigar form is less chemically bound to the substrate. We can analyze the amino acid in contact with gold in the similar way we have done with the pH. In the cigar shape, we are able to detect not only the tryptophan and the lysine, but also the tyrosine. We observe again that the cigar shape is less favorable in terms of protein absorption compared to the native shape. Nevertheless, the amino acids are attached to gold without ionic bridge, which enhance the stability and surface affinity. In the denatured form at 50 degrees Celsius, we detect both the hydrophobic amino acid and also the phenylalanine. The thiol group was also visible. All this gives a stronger absorption than for the cigar shape. To conclude, we hope we have convinced you of the interest of single protein measurements for determining the secondary structure of protein. Counting the spectra enables to determine the structure transition with a better accuracy than a fitting procedure. Controlling the environment of the protein, such the pH, the temperature, the surface, is of fundamental importance to orientate the protein and maintain the stability of the protein structure. We found the gold surface does not impact the secondary structure. However, the electrostatic properties of the surface, thiol group and citrate agent are responsible of the orientation of the protein toward the surface. Perspective of this work is to investigate the naturally disordered protein. Thank you for your attention.